Variety, it is said, is the spice of life. And in living organisms, variety arises through mutation. The amazing diversity of life, red hair, opposable thumbs, speech, sunflowers and squirrels. These are a few of my favorite things. And those, along with many other features we take for granted, arose through mutation. If it weren't for mutation, life on Earth would be exactly as it was when the first cells arose. With no change in the genetic information, there could be no change in the kinds of organisms that exist. Mutations are said to be the raw material of evolution, the variations in a population that make it more likely that there will always be more individuals that can deal with unexpected changes in the environment. Monoculture, the practice of growing a single variety of a crop on a large scale, is dangerous. A pest or disease can wipe out every plant for miles around if the plants are genetically identical. Conversely, outcrossing, that is, breeding with a different population, infuses variant genes into a group and is often accompanied by an increase in hardiness or hybrid vigor. Mutation, it seems, is necessary and useful. And yet cells go to an enormous amount of trouble to prevent mutation. You already saw in the lectures on DNA replication that DNA polymerases check their work and correct their mistakes, reducing the error rate to as low as 1 in 10 million nucleotides copied. But proofreading polymerases are just the beginning of what allows us to copy 6 billion nucleotides accurately and defend against unwanted changes or damage to the information in our DNA. The 2015 Nobel Prize was awarded to Thomas Lindahl, Paul Modrich, and Aziz Sankar for unraveling 3 major pathways for DNA repair. These mechanisms work ceaselessly to maintain the integrity of the genome and ensure that information passed on to the next generation is a faithful copy of the original. In this lecture, we will examine the numerous mechanisms that cells employ to minimize alterations to DNA, which is the master copy of instructions for an organism. But first, let's start by examining the ways in which information in DNA can be distorted or lost. Considering the sources of mutation and damage to DNA will give us a sense for the magnitude of the problem. Information can be degraded through mutation, that is, changes in the DNA sequence, or by damage to the DNA molecule. One way that DNA sequence can be changed is if there are mistakes made while replicating DNA. And while proofreading by DNA polymerases reduces the incidence of such errors greatly, not all mistakes made during copying are caught and fixed. While 1 mistake in 10 million is better than human typists, remember that replication in our cells entails copying 6 billion nucleotides in each round. At that rate, there could be 600 uncorrected mistakes per round of replication in each cell, even with proofreading. Now that starts sounding much more serious. And when you factor in the trillions of cells in the body that are dividing at any given time, the total number of potential errors becomes truly staggering. Now add to that the fact that even when DNA is not being replicated, it suffers damage over time. Sources include sunlight with wavelengths in the ultraviolet range and cosmic radiation from the sun and stars. Radioactive elements such as radon are another source of damage. And if we need medical diagnostic procedures, X-rays and CT scans add to our radiation exposure. Chemicals in the environment also cause damage to DNA. Mutagenic compounds are found in tobacco smoke and auto exhaust, as well as in such apparently innocuous things as grilled meats. Even chemical reactions naturally occurring within cells give rise to compounds that can damage DNA. Add it all up and it starts to seem positively amazing that we are not mutant monsters. And in truth, if the mutations and damage weren't corrected, we would be in a very sorry state. In fact, defects do sometimes get past our repair systems, and when that happens, various kinds of cancer and a slew of other health issues can result. So we really owe our continued functioning to the numerous repair systems cells use to ensure that our DNA is kept in good working order. How do these systems work and what exactly do they fix? Let's begin by looking at a cellular system for correcting any replication errors that were missed by the proofreading function of DNA polymerases. The integrity of information in DNA is compromised when DNA polymerases incorporate the wrong nucleotide into the new strand and the proofreading system does not detect it. Now, how might this happen? One way is by transient formation of isomers of the bases. What happens is some of the atoms mediating hydrogen bonding temporarily change position and stabilize base pairs that do not normally form. For example, G and T normally exist in the keto form with double bonded oxygens characteristic of ketones. But they can sometimes isomerize to the enol form with an OH characteristic of an alcohol. Similarly, C and A normally exist in the amino form, but may sometimes take on the imino form of a C double bonded to nitrogen. These rare isomers have different base pairing properties, permitting isomer C to pair with A and isomer T with G. These are mispaired nucleotides. They won't convey correct genetic information, but they do get incorporated into the new strand of DNA that is being built. And they do not get removed by the DNA polymerase's proofreading activity that checks for incorrectly paired bases, since the isomer base pairs appear legitimate for the brief time that they isomerize. The isomers are transient, lasting no more than seconds, but it's enough to fool DNA polymerase. Insertions and deletions can also occur when DNA replication occurs in regions of short repeated DNA sequences. These can happen as a result of one strand sliding with respect to the other. Errors not caught by proofreading during replication can be corrected by a mechanism called mismatch repair. While the error rate of DNA replication is about 1 in 10 million base nucleotides in the absence of mismatch repair, in the presence of mismatch repair, this rate is further reduced better than 100-fold to fewer than 1 in a billion nucleotides. Let's think about what such a system must be able to do. First, it must scan the newly made DNA to see if there are any mispaired bases, for example, a G across from a T. Then it must identify and remove the nucleotides in the region of the mismatch. And finally, it must correctly fill in the gap created by the excision of the mismatched region. Crucially, the mismatch repair system must also have a means to distinguish the newly made DNA strand from the template strand if replication errors are to be fixed correctly. For instance, when the mismatch repair system encounters an AG mispair, it must know whether the A should be removed and replaced with a C, or if the G should be removed and replaced with a T. If the mismatch repair randomly fixes problems, it'll be wrong half the time. So how does the mismatch repair system distinguish between the original and the new strands of DNA and improve the error rate? E. coli, for example, has an enzyme called DAM, short for DNA adenine methylase, and it adds methyl groups to adenines in the GATC sequences of DNA. The sequence GATC reads the same way in the strand complementary to it. So you can see here the top strand has a G with a methyl A, followed by a T and a C. And the bottom strand, if we read it in the 5' to 3' direction, also reads G, followed by a methyl A and a T and a C. So both adenines in this sequence are methylated. When DNA replication copies one of the strands, the newly synthesized GATC will not be methylated at first, as this requires remethylation by DAM to put the methyl group onto the adenine in the new strand. Until the methyl group gets put on, the GATC sequence is said to be hemimethylated. The old strand is methylated and the new strand is not. The mismatch repair system in E. coli appears to assess the methylation status of GATC sequences nearest the mismatch or error. Newly made DNA has not yet undergone methylation and thus can be distinguished from the template strand, which is methylated at all GATC sequences. The mismatch repair proteins selectively replace nucleotides in the strand lacking methylation, thus ensuring that it is mistakes in the newly made strand that are removed and replaced. In E. coli, mismatch repair proteins are encoded by a group of genes collectively known as the mut genes, short for mutation. Important components of the mismatch repair machinery are the proteins mut-S, mut-L, and mut-H. Mut-S's role is to flag the mutation by binding to it. This causes a bend in the DNA that serves as a signal for the binding of the other proteins. The second protein in the mismatch repair is called mut-L, that then binds to the first protein to serve as a bridge for the third protein. Mut-H then cuts the newly synthesized DNA strand, which is not yet methylated, making the cut at a GATC. This activates a DNA helicase and an exonuclease that help to unwind and remove the region containing the mismatch. DNA polymerase III fills in the gap using the opposite strand as the template, and ligase joins the ends to restore a continuous strand. This may sound a little like the way Okazaki fragments got filled in during DNA replication by DNA polymerase I. However, repair is different, and the repair process here is not occurring at a replication fork. Eukaryotes also have a mismatch repair system that repairs not only single base mismatches, but also insertions and deletions. Proteins corresponding to the E. coli system of mut-S, mut-L, mut-H have been identified in other organisms, including human beings. These, together with additional proteins, carry out mismatch repair in eukaryotic cells. However, eukaryotes do not use DNA methylation to distinguish the new strand from the old. Instead, there's evidence that the newly made DNA may be recognized by the fact that it is nicked or discontinuous. The presence of discontinuous strands due to Okazaki fragments that have not yet been joined together may permit the new strand to be distinguished from the older, continuous parental strand. In summary, the mismatch repair system makes a 100-fold improvement in accuracy or fidelity of the replication process. Not surprisingly, defects in mismatch repair can spell trouble. In addition to leaving in mismatch bases, mismatch repair deficiencies can also lead to uncorrected errors in the replication of short repeated sequences in DNA. Failure to handle short repeated sequences is called microsatellite instability. This problem is usually prevented by functional mismatch repair, which can fix not only single base mutations in DNA, but also small insertions or deletions of sequences during replication. When that ability is impaired, the repeated sequences vary in the number of repeats that each strand contains. Such variations are associated with a number of disease conditions. For example, mutations in human mismatch repair genes are associated with the development of Lynch syndrome, also known as hereditary non-polyposis colorectal cancer, or HNPCC. People with Lynch syndrome have an 80% lifetime risk for colon cancer. They also have an elevated risk for other cancers, including those of the breast, prostate, bladder, and thyroid, among others. So failure of mismatch repair has pretty severe consequences. But even with a perfectly good mismatch repair system, we cannot escape DNA damage. DNA is vulnerable even when it isn't being replicated. One source of damage that surrounds us is exposure to UV radiation in sunlight. The same sunshine that lifts our moods and helps us make vitamin D in our skin can also wreak havoc on our DNA. UV radiation can do its dirty deeds even on cloudy days, but the damage is naturally much worse on a clear, sunny day when more of the radiation reaches your skin. On a day when you're lying out on the beach, your skin cells are readily accumulating damage to your DNA. This, however, is not the source of your misery when your skin turns red, hot, and painful. Those symptoms are the result of inflammatory responses and increased blood flow to the skin in response to the assault on your DNA. What is more worrisome than the discomfort of a burn is the pileup of DNA damage that will need to be repaired. Cells with extreme levels of damage will be triggered to undergo programmed cell death so that they do not proliferate, which is why your skin begins to peel. The dead cells are being sloughed off. Meanwhile, repair systems will do their best in the remaining cells. The real risk is that repair systems may be overwhelmed or that not all cells with excessive DNA damage will die. Proliferation of damaged cells can lead to skin cancer. Now, what sort of damage does UV do at the molecular level? One common form of UV damage is when two adjacent pyrimidine bases, that is, Cs or Ts, get cross-linked in a single strand of DNA. Again, we're talking about two neighboring pyrimidine bases linking up on the same strand of DNA. These linked up pairs form what are called cyclobutane pyrimidine dimers, or CPDs. Another way for UV exposure to damage Cs or Ts is for the 6 carbon of one ring to bond with the 4 carbon of another, forming what's known as a 6,4 photoproduct, or 6,4 pp. Both of these types of mutations distort the DNA helix, pulling it out of shape and blocking replication and transcription. It should be obvious that if the UV in sunshine damages your DNA, the same wavelengths are equally harmful when they come from a tanning bed. Contrary to claims, mostly by tanning salons, that their tanning booths are safer than a sunburn, they will damage your DNA just as much, if not more. Using sunscreens or sunblocks that reduce the UV radiation that reaches your DNA is helpful. And being naturally dark-skinned provides some measure of protection because higher levels of melanin pigment in dark skin shields DNA from UV rays. But nothing can prevent a certain amount of damage that will need to be repaired by your cells. Cells have a whole set of proteins that deal with UV-damaged DNA. This system, called the nucleotide excision repair, or NER system, also fixes damage from environmental chemicals such as those found in tobacco smoke, automobile exhaust, etc. These carcinogenic chemicals include molecules like dibenzopyrene that can attach themselves to bases. These attachments of large chemical groups to bases in the DNA are referred to as chemical adducts or DNA adducts. Like the damage caused by UV radiation, chemical adducts can physically distort the DNA helix, causing DNA and RNA polymerases to stall when they attempt to copy those regions of DNA. Nucleotide excision repair proteins cut the damaged strand on either side of the base that's been chemically altered, called a lesion. A short portion of the DNA strand containing the damage is then removed, and a DNA polymerase fills in the gap with the appropriate nucleotides. Nucleotide excision repair has been extensively studied in bacteria. What happens is the damaged strand is recognized and nucleases excise it while leaving its partner strand intact. The gap where the DNA was gets filled in by DNA polymerase and ligated together by DNA ligase. Nucleotide excision repair is also important in eukaryotes. A number of proteins have been identified that function in ways similar to the bacterial repair proteins. The importance of these proteins is evident from the fact that mutations in the genes that encode them lead to genetic diseases. For example, Cockayne syndrome is characterized by severe impairments including microcephaly, hearing and vision loss, and extreme sensitivity to sunlight. Like those with Cockayne syndrome, people with xeroderma pigmentosum, or XP, are very sensitive to UV exposure. The damage caused by UV radiation cannot be repaired in these individuals without a functional nucleotide excision repair, leaving them at a 1,000-fold higher risk of developing skin cancer than the general population. Indeed, children with this condition may develop their first skin cancer by age 10. Lifestyle interventions like scrupulously avoiding sun exposure and consistent use of high SPF sunscreen can help reduce the damage. More recently, creams containing DNA repair enzymes have been formulated to apply to the skin of XP patients. These show some promise in helping mitigate the consequences of accumulated UV damage. One reason that humans are peculiarly susceptible to the harmful results of malfunctioning nucleotide excision repair is that it is our only system for dealing with UV-induced lesions. In this respect, we are woefully inadequate. What about plants? Plants are exposed to UV on an ongoing basis and obviously cannot move themselves out of the sun. UV damage is inevitable, but it turns out that plants have a very efficient system to deal with the mistaken cross-linking of C and T bases induced by UV exposure, thanks to enzymes that can directly reverse the mistaken cross-linking. These enzymes, called photolyases, are able to use the energy from blue wavelengths of light in sunshine to undo the crosslinks created by the UV light. So even as UV exposure causes the formation of crosslinks, the blue light wavelengths are used to immediately and efficiently remove them. Pretty nifty. Plants also have a nucleotide excision repair system to back up their efficient photolyase. Now, in a cruel twist of fate for humans, it turns out that even many other animals, from birds to marsupials, appear to have photolyases. It's only humans and other placental mammals that lack the enzymes to repair UV damage. Anyway, because we rely so heavily on our nucleotide excision repair system, we're much more likely to suffer from the effects of unrepaired UV damage. And we're out of luck if our nucleotide excision repair does not work. But guess what? That may change in the future. Scientists have successfully transferred the photolyase gene from a plant into mice and shown that it works to repair UV damage in the mice. The mice with the plant gene for photolyase were able to withstand doses of UV light that caused severe sunburn in normal mice, and they also had greater resistance to developing UV-induced skin cancer. Another study in 2014 showed that human skin cells could be made to produce the photolyase enzyme from the South American marsupial opossum by introducing messenger RNA and coating the protein into the cells. Once made in the skin cells, the enzyme efficiently cleared UV-induced damage from the DNA of the cells. If improved methods to deliver RNAs into cells are developed, this could alleviate the suffering of people with conditions like XP. But there are other sources of genetic mutation besides environmental insults, like radiation or chemicals. DNA can also be damaged by events inside cells. For example, the DNA base cytosine is relatively unstable chemically and can sometimes spontaneously lose an NH2 amine group and convert to uracil. Other sorts of damage result from the normal chemistry of the cell. In the lecture on free radicals and healthy eating, you encountered reactive oxygen species, or ROS molecules, that are highly reactive because of their unpaired electrons. Such reactive species can react with guanine, for example, leading to an oxidized form of guanine called 8-oxoguanine. Methyl groups and other short carbon chains can be added to bases by chemicals called alkylating agents. For example, a methylated form of guanine might get a methyl at the O6 location to become what is called O6-methylguanine. All three of these modified bases do not actually change the physical structure of the DNA helix, but they can cause problems. What happens is the modified bases may end up with incorrect pairings compared to the unmodified forms, leading also to mutations in the next round of replication and corresponding bulges in the DNA duplex. For example, cytosine would pair with guanine, but if cytosine converts into uracil, then that uracil, if unrepaired, would instead pair with adenine. Similarly, the methylated form of guanine, O6-methylguanine, doesn't behave like guanine in pairing. It pairs with thymine. Even more bizarre, the oxidized form of guanine pairs with A, even though both A and G are purines. For this reason, individual bases that have been modified or altered must be removed and replaced by repair systems. There are two ways this can happen. The first of these is direct reversal of the modification. O6-methylguanine in DNA can be removed by the direct reversal method with the help of a very unusual enzyme. This enzyme removes the methyl group from the guanine and transfers it onto a cysteine residue in the enzyme. The addition of the methyl group to the cysteine also renders the enzyme non-functional. That is, O6-methylguanine methyltransferase, the enzyme, is a single-use enzyme. Fix one O6-methylguanine and the enzyme is kaput. Of course, true enzymes are catalysts that remain unchanged over the course of the reaction. So to emphasize the contrast, this kind of catalyst has been called a suicide enzyme because the enzyme dies as a result of its own activity. Another repair system for individual bases is called base excision repair, or BER. It can remove modified bases like 8-oxoguanine as well as uracils derived from cytosine deamination, and is yet another multi-protein repair system. However, base excision repair functions a little differently than nucleotide excision repair. In base excision repair, a single damaged base is first removed from the DNA, followed by a removal of the region of the DNA surrounding the missing base. The gap is then repaired. The removal of uracil from DNA is accomplished by an enzyme that can recognize uracil in DNA and break the bond between the uracil base and the sugar in the nucleotide. Removal of the base leaves a gap where the uracil, a pyrimidine, was removed. That is, the DNA is lacking a base in the middle of the double helix, kind of like a tooth that's been knocked out. So the gap is called an apyrimidinic site, or an AP site. Formation of the AP site triggers the activity of an enzyme known as AP endonuclease. This endonuclease enzyme cuts the DNA strand containing the gap at the AP site. This cut removes that part of the strand where the tooth is missing, while leaving the other strand intact. It's similar to what happened in nucleotide excision repair. Then a DNA polymerase binds to the gap and replaces the missing sequence. Finally, as always, DNA ligase acts to seal the nick in the DNA. So we've seen that there are several different kinds of excision repair, but they all involve cutting out the portion of the DNA that's damaged, followed by repair synthesis using the other strand as a template, and finally a ligase enzyme to restore continuity to the repaired strand. Cells possess several different kinds of excision repair, each geared to specific kinds of DNA damage. Between them, these repair systems deal with the wide variety of insults to the genome. Although we think of defects in DNA repair mechanisms as detrimental to the health of the organism, there are circumstances where the opposite might be true. Radiation therapy and drugs used for chemotherapy work by causing extensive damage to DNA in cancer cells. The idea is to damage the DNA of the cancer cells sufficiently to trigger programmed cell death or apoptosis. However, if the DNA repair systems in the cancer cell are robust, the cancer cells may survive. And to ensure that the cells are killed by these therapies, one strategy is to dial down the DNA repair systems using drugs that inhibit them. Cancer cells are sometimes prevented from surviving chemotherapy by inhibiting the AP endonucleases that cancers normally use in their own base excision repair. An inhibitor drug known as leucanthone, for example, inhibits one of the AP nucleases. In treatment of brain cancers, it is sometimes given in combination with radiation, since the radiation damages DNA in the tumor cells. Leucanthone is a drug whose ability to traverse the blood-brain barrier makes it an important treatment for this kind of cancer. Gene repair can do great things and also do great harm. It's important to understand both. Gene repair can be our best friend when it fixes damage to DNA in our healthy cells or prevents mutation that could lead to formation of a tumor. On the other hand, gene repair can be our worst enemy whenever cancer cells survive our treatments by repairing their DNA faster than we can inhibit and interfere with those repair efforts. In the next lecture, we'll see how the understanding of gene repair has contributed to powerful technologies for gene editing.